A very good morning and good afternoon to everyone, depending on where you're joining us today. My name is Joy Khan, and I'm the Regional Marketing Manager for Asia Pacific from LGC Biosearch Technologies. Before we introduce the topic and speaker for today's webinar, let us just go through some of the housekeeping rules first. So all participants will be muted to prevent background noise during the presentation, and I hope you understand that. And we have prepared a few polling questions during certain breaks of this webinar, and we really appreciate and encourage you to participate, and we're keen to understand what are your research areas, what might be some of your techniques that you're interested, and how can this single molecule RNA fish technology be utilized to achieve your project goals. And after the webinar, we will have a Q&A session. But you're always welcome to enter your question in the question window at any time during the event. We will try to answer as many questions as possible during the Q&A session, but do rest assured that any unanswered questions that we don't have time for today, we will be able to address them and deliver them to you via email or via some other channel. And after the event, we will also send an email with a link that you can access for the recorded event after this webinar. And once again, thanks for joining us today. So with that, let me start on the next slide, and we will introduce on the topic as well as the speaker for today's webinar. So today's webinar is on expert tips to generate publication-ready single-cell spatial gene expression data with this exciting technology called Stellaris RNA fish technology. And our speaker for today is Dr. Julie Mangada. Julie is the head of training and technical support from LGC Biosearch Technologies. Dr. Julie Mangada specialized in immunology, where her doctoral research at University of Massachusetts Medical School worked to tease apart the molecular mechanisms of autoimmunity and how they relate to transplantation tolerance induction. She then joined the Buck Institute for Research on Aging as a postdoctoral researcher, working with human embryonic stem cells and their differentiation into neurons for cell replacement therapies. These studies led to the development of a mRNA-based imaging platform for studying transplanted dopaminergic neurons during the peri-transplant period in a Parkinson's disease model. She then joined LGC Biosearch Technologies back in 2016 as head of training and technical support, where she really has vast experience helping our global Stellaris RNA fish customers to get started with their projects, of course, among other portfolios that she supports, such as oligos that's being used in technologies like polymerase chain reaction, PCR, as well as next generation sequencing, NGS. So with that, I'll pass on to Dr. Julie Mangada to give us the webinar for today. Dr. Julie, please.
Thank you so much, Joy. I just want to start off by saying hello to all of you that are on the line and give you my heartfelt gratitude for your interest in Stellaris RNA fish. This is actually one of my favorite product portfolios that my tech support teams support, and really excited tonight to be talking about something that's not related to a SARS-CoV-2 primer or probe. So thank you all so much for your interest. So we'll go ahead and get started. And I'm going to start off by just throwing up some trigger words. These are, I think, a great example of the breadth and scope of Stellaris RNA fish. It's a really cool chemistry and an exciting imaging technology, and it generates some pretty incredible data and pictures, publication-worthy pictures. So I'd just like to kind of start off by highlighting some of the many different applications of Stellaris RNA fish. First of all, starting off with, of course, the keyword mRNA. Stellaris RNA fish is a fluorescent in situ hybridization for mRNA, but not just mRNA. It's also great for detecting long non-coding RNAs. I can think off the top of my head, two very popular ones we have are the mallet1 Stellaris set, which is specific for long non-coding RNAs associated with specific types of lung cancer. We also have a very popular set for NEAT1. NEAT1 long non-coding RNA expression is changed by HIV infection. The next trigger word, of course, is microscopy. And microscopy, I'm talking about wide field microscopy. Fluorescent wide field microscopy, that's where you're going to have the most of your success with the Stellaris fish imaging technique. A lot of people love their confocals. It's just a little more difficult for reasons I'll get into a little bit later, but wide field fluorescent microscopy is really the work engine for this chemistry. Then we're getting into immunofluorescence, or IF. One of the coolest things about Stellaris RNA fish is that when you couple it to immunofluorescent techniques, you're actually able to simultaneously detect mRNA and protein at the single cell level. Talk about spatial and temporal regulation of not just the transcript, but the translated protein. So those are some really great images to look at. Knockdown verification. We have a lot of customers that use Stellaris to verify knockdown of a specific transcript or gene of interest. So any of you that are working with transgenic cell lines, this is a great control to have in your toolbox. Cell-to-cell variation in gene expression. As many of you know, at the cellular level, neighboring cells can have vastly different expression profiles, especially cells that are in a disease state or cells that are differentiating. Also, what comes to mind is the generation of induced pluripotent stem cells from somatic cells. So having the ability to track gene expression and upregulation or downregulation at the cellular level is pretty important when you're studying IPS or cancer or some of your other cell lines. Ice fish. I'm not sure how many of you have heard about the ice fishing technique, but it's actually a really great application for Stellaris because, well, as you know, most post-transcriptional processing, including pre-mRNA, occurs co-transcriptionally. So RNA fish targeting the location of introns can actually be used as a proxy for the encoding gene. That's what ice fishing is. It allows you to use the Stellaris fish method as a functional proxy for DNA fish because Stellaris RNA fish never directly targets DNA. Our protocols and probes aren't set up for that. But with ice fishing, you can make a lot of empirical determinations about the co-transcriptional modification and expression. Localization and co-localization. Stellaris can be used to determine where very unstable transcripts are actively transcribed or illuminate a lot of your important mRNA and protein interactions. So you get a lot of data about localization and co-localization, especially for unstable transcripts that don't hang around in the cell for a long time. Cell type specific expression. So here again, I'm going to bring to mind induced pluripotent stem cells or cells that are in an altered disease specific expression state, like your cancer target cells. Something else that comes to mind for cell type specific expression, I'm thinking about some studies that came out of MIT a few years ago. If any of you are familiar with an imaging technique called Clarity, Clarity has used Stellaris in a publication that utilizes whole tissue clearing technology, getting rid of the lipid bilayer of the cells and allowing for visualization of RNA distribution and antibody staining in an entire 3D intact mouse brain. So that was a really exciting application for Stellaris, and was able to show in the brain, in their own native niches and zones, the differential expression of different cellular receptors and signaling molecules. Antibody validation. A lot of our customers use the Stellaris technique to validate their antibodies. They can use the positive signal from Stellaris to validate the fact that their antibodies are indeed specific. Splice variants. People use Stellaris to really interrogate either all or most RNA variants from one gene. That would be an inclusive set. Or if you're designing an exclusive set, you can target either a single or just a couple RNA variants from the same one gene. If you design separate Stellaris sets, some would be inclusive, some would be exclusive, you can actually combine that data to further define actual variant copy number and ratios. Downregulation and upregulation. Again, Stellaris can be used to determine the changing tempo of expression in response to either a disease state or drug treatment, or just a cell going through its normal differentiation program. RNA processing. Stellaris is used to help understand the regulatory mechanisms of gene expression, RNA processing, and splicing all together in one experiment. And cell lineage, finally. You can use Stellaris staining to actually characterize the plasticity of, say, pluripotent stem cells and follow their different expression profiles as they go from that single progenitor cell all the way through into either mesoderm, endoderm, or ectoderm, just as one example. So hopefully I've highlighted and convinced you that Stellaris RNA fish has a wide variety of applications and is a pretty exciting technology to use if you want some great publication-ready pictures for your studies.
Great, thanks Julie for the overview of the applications, what might be useful for you. And we now come to the first poll question of this webinar. We're keen to understand, what is your research field? Please select all that apply. Are you in cancer biology? Are you working on neuroscience? Or are you an immunologist? Or you might be working on others. So feel free to take your answers, and we'll be sharing with you the survey results in real time in a few more seconds. Great, we have the results now. Seems like we have quite a number of attendees here who are doing other field research, and we like to hear from you. If you feel comfortable, feel free to type in some of your research specific questions in the chat box, and we'll be able to address them towards the end of this webinar to see how Stellaris might be able to help with your research. But the second, we do have quite a number, about 37% of the attendees are working on cancer biology, followed by 24% being immunologists, and remaining 1/5, 20% are neuroscience. Great. Thanks for your participation, and let us move on to hear more about the Stellaris from Dr. Julie Mangada.
Yeah, thank you. That is really illuminating to see the different areas people are using this technology. So I'm thinking most of you are pretty familiar with the Stellaris technique and what it does, but I don't want to make that assumption and lose somebody if they're not completely familiar. So I'm just going to start by introducing some basic foundational knowledge about Stellaris, and then we'll move on into some of the more specific applications. So here, what I'm showing is that classic cartoon that depicts what they've always called the central dogma of molecular biology, which is where you go from DNA to RNA to protein. Now, of course, since the burgeoning field of epigenetics has taken off, we understand that while this is a very important flow of information from DNA to RNA to protein, it's certainly not the only information flow. But generally speaking, going from DNA to RNA to protein, RNA is right in the middle of that cellular conversation to get those proteins expressed and regulated. So typically what would happen is if you were studying RNA and you wanted to know which RNA is being expressed at a specific time, you would look at either microarray or RNA-seq data, and you see an example of that data on the slide. So when you're asking which RNA is expressed, microRNA or RNA-seq, but if you want to know where that RNA is being expressed, then you have to do a different set of questions. And usually that involves either immunofluorescence or immunohistochemistry. And this picture here is actually a gorgeous example of some immunohistochemistry staining in mouse brains looking at organization and marker expression of different progenitor zones in cortical neuron subtypes. So you can see they're getting a lot of information about where those expression markers are being expressed. And then if you wanted to ask how much is being expressed, so now you want to know an actual quantifiable data point, you would have to do either northern blot, which was semi-quantitative, or you would do qPCR. And this is just an example using our own IntelliCube, spiking in human genomic DNA, just showing the dynamic range that goes from 4 times 10 to the 7 all the way down to just 40 copies per reaction. And trying to get all of that information together where it's quantitative, how much is expressed, where is it expressed, and which RNA is being expressed. Again, you would be utilizing all of the different modalities. But with Stellaris RNA fish, you can actually answer all of those questions in one experiment because Stellaris, once you target a transcript and generate your probe sets, you'll know exactly which RNA is being expressed. You'll know in an intact cell and even in multiple neighboring cells, if you're looking at tissue samples, you'll know where it's expressed. And because it is quantitative, you're going to be able to ask questions at the single molecule level. How many molecules of that mRNA are there? So how is this technique accomplished? It's actually pretty straightforward. I'm sure most of you have experience with immunofluorescence and immunohistochemistry, where you're using primary antibodies, secondary antibodies, maybe colorimetric assays with the deposition of different enzymes as your readout. And a lot of those protocols actually take a couple days. By the time you're finished adding your secondary and then curing, you can be 2 or 3 days into the protocol. Really nice thing about Stellaris is actually the quickness in which you can do your protocol. There's not a whole lot of steps to it. In fact, it's pretty straightforward. As you can see, looking at the top panel, that is showing just the time course. And a lot of Stellaris techniques can be started, completed, and imaged within one laboratory working period. And if you look at the bottom panel, it's really pretty straightforward. There's no exotic reagents that are required. Here I'm showing a cartoon of a cell, and to that cell, after a fixation, permeabilization, washing, you add the wash buffer, you're going to add your individually labeled oligos. There's a short period of incubation. Those oligos, upon seeing their targeted transcript, will actually tile along that transcript. You then treat the cells with an anti-fade and a DAPI stain, put on your cover slip, and then you're ready to image with the microscope. So it is actually a very simple protocol. Now, what that looks like is this. Again, this is just a cartoon representation, but as you can see, I'm depicting a squiggle, which is actually your RNA transcript, and each one of those fluorophores is a probe. The probes are lining up along that RNA transcript, and when you go to image them with the microscope, you're going to see a single punctate signal that's actually the aggregate of all of those fluorophores that are lined up, anywhere from about 25 to 48 individually labeled oligos tiled along one mRNA transcript will show up because of the diffraction limit. Dots that are within 200 nanometers will merge as one single punctate signal. And that is the real power in Stellaris, because what I'm describing here, what you see in this cartoon, there's no amplification step. So a Stellaris signal, that single punctate dot, is the aggregation of multiple fluorophores due to their diffraction limit showing up as one signal. It's not a strong signal, but it's not amplified. It's not a false signal, and that's what allows Stellaris to be quantitative. So Stellaris, through the years, has actually been growing in popularity. If you go to our website, you can actually see we have a landing page, the Stellaris Citation Center, and it'll actually break down and show you all of the different publications that Stellaris is currently in. And something that I find as a scientist very rewarding is when you look at the impact factor of a lot of the journals that Stellaris is in, even making the cover of these journals, these are a lot of high impact journals like Nature, Cell, and Science. We have another polling question.
Yes, thanks, Julie. Now we have come to the second polling questions. We are keen to understand what sample types do you normally work with. Do you work with adherent cell lines, or is it FFP samples? Is it frozen tissue, whole month samples, or others? Feel free to put your answers here, and we'll be sharing the answer in a few seconds. All right, great. Seems we have almost half of the attendees here are working with FFP samples, followed by adherent cell lines, about 41%, followed by frozen tissue, 1/3 of them, about 32%, and about 1/4 are working on whole month samples, and a few others are working with other sample types. So if you do find some of your sample types to be of rarer type and you would like to understand how Stellaris might be able to work on those sample types, feel free to put your questions in the chat box and we'll be able to address them towards the end of this webinar. All right, let's go ahead. Dr. Julie.
Thank you. That actually is really surprising to me. I would have predicted most were working with adherent cell lines. So thank you. That's really good information to have, actually. Um, so now we're going to get into a little bit of the basics of designing your own Stellaris experiment. And these are things, regardless of the sample type you're working with, you're still going to be following the same um, background of choosing your targeted region, how to design your probe sets, choosing the appropriate positive and negative controls, and then it's not until you get to the actual imaging that you're going to have careful consideration of which protocol to use. So we're going to start off with designing a Stellaris probe set. And I'm putting this up here just as an example that when you're designing your set, understanding that these are for research use only, this is not a high throughput multi-platform screening paradigm for clinical samples. We're talking about research use only. And I did mention it a little bit in the beginning, but you have the ability to design inclusive versus exclusive probe sets. Now, by inclusive, I mean all or most RNA variants of that gene. By exclusive, I mean you're targeting either a single or just a few RNA variants of that same gene. So you can combine them to get all kinds of information on copy number and ratios. Now, if you want to detect mature RNA, then we recommend designing your set against the coding sequence if possible. And that all depends on how long your coding sequence is. We do recommend that Stellaris probe sets have at the minimum 25 individually labeled oligos in order to generate the aggregate of enough signal to get that one punctate dot that you're going to be able to count in the software. So if your targeting region isn't long enough for those probes, which are individually between 18 to 22 nucleotides long, then you're not going to be able to generate those minimum required 25. So the length of your transcript has a lot to do with it as well. For short mRNA sequences or RNAs from genes with very similar homologs in the organism that you're actually testing, we strongly recommend careful upfront bioinformatic analysis, doing all of that groundwork ahead of time. That means blasting and multiple sequence alignments to better determine if 5' and or 3' untranslated regions are suitable for inclusion in the design. We also recommend a manual blast of each individual probe that the probe designer generates. And if there's a masking level of 3 or below, you're going to want to be very rigorous with blasting the probe sets that are generated against that organism and even against other organisms. So in that case, with a masking level of 3 below, and by masking level, I mean when you're designing your probe set, the masking level of 5 is the most rigorous. That's going to exclude any potential cross hybridization with the off targets. Masking level of 3 and lower is a little bit more promiscuous with off target binding. And in that case, you would want to exclude any 20-mer oligos that have greater than 16 nucleotide matches to other RNAs to minimize the possible cross hybridization, especially if these multiple probes are going to bind off target. And remember, because it's the aggregate of at least 25 individual probes tiled along one transcript, off-target binding of a handful of probes will never confound your results because it won't generate a signal. So when you're blasting the probes that the designer generates, you're actually looking to remove more than half of the probes if they're going to be off-target binding. Just a few off-target binding won't generate a signal. Also keep in mind when you're designing these probe sets that the probe designer takes into account and balances both GC content and the TM. And that GC content and TM is specifically meant to be used with the protocols that we've developed for the Stellaris technique. And I did mention, but it bears mentioning over and over again, that for single molecule detection, to really make this quantitative, we recommend there be at least 25 individual probes in the set that you order, while staying within the maximum limit of 48 probes per probe set. So now we're going to talk a little bit about the microscope. In my experience supporting Stellaris, I run the team. All of these Stellaris questions that come straight to my team. And in my experience, the most important part of experiments using Stellaris RNA fish probes, not to mention having an RNase-free workspace, but it's actually proper image acquisition and deconvolution of the obtained Z-stacks. So having the appropriate microscope, which, as I mentioned in the beginning, we recommend a wide field microscope. That being said, I guess I should caveat this with, while highly experienced RNA fish and confocal microscopy users have experienced successful Stellaris RNA fish imaging, we actually don't recommend the use of confocal microscopes, especially not as an entry point if you're just learning the Stellaris technique. That's basically because this type of microscope, the confocal, uses point illumination to limit the focal plane for imaging and restrict out of focus light, but that actually diminishes the sensitivity of what's already a low light imaging chemistry. So if you can, we recommend having that wide field fluorescent microscope. The next thing that you want to keep in mind is your filter set. You're always going to want to make sure you're choosing dyes that are compatible with the filter cube of your microscope. If you have any questions, you can write into tech support. You'll see our email at the end of this webinar, and we can get the part numbers from your microscope filter cube and tell you which of our fluorophores are the most likely to work with your specific detection limitations. I can say that Quasar 570 and 670, which align closely with the excitation and emission spectra of Psi3 and Psi5 respectively, are among our most commonly used fluorophores. That's the ones that most of our customers use in their publications. You also really want to pay attention to your eyepieces and your camera and the lamp that you're using. That's because, we do get this question. You're not going to be able to view most fluorescent RNA spots using the microscope eyepiece. So it's essential to have a proper light source and camera. You definitely want to acquire images. I had mentioned, in our experience, the most important part of this technique is the image acquisition and deconvolution of Z-stacks. You absolutely have to acquire images using 1 to 2 second exposures per plane with about a 0.25 micron in between Z sections. And then you can go into your post-acquisition software and perform maximum intensity projection. We use software like ImageJ. I'll go over some examples of a couple different software here on this slide. Of course, there's Star Search, which is a great spot counting tool. Also, I mentioned ImageJ. That's one of the big ones that we use here in the lab. There's lots of others on this list. The ones that I've listed here are actually free. You can download them. There's others that I believe have a fee, like Metamorph and Elements, but all of those would be perfectly acceptable for your deconvolution software and to work with your data after image acquisition. So now we're going to talk a little bit about the spot counting. Of course, this is what makes Stellaris quantifiable, where you can actually make claims about very specific data as far as levels of mRNA expression at the single molecule level. The one that we use a lot is called Star Search. You can download that online. I've put the link here from the developers lab at UPenn. He's one of the collaborators and the inventor of the Stellaris technique. This particular program, the Star Search program, is well suited for single molecule RNA fish, and it uses those Z stacks that I mentioned as an input. It can even distinguish two overlaying spots and count them separately. So having good data analysis, acquisition, deconvolution, and then quantification is critical. So now once you start jumping into your Stellaris experiment, it's going to be really important to actually verify your probe set. You're going to want to convince a reviewer of any sort of publication that your probe set is both specific and sensitive. So one of the common things that we use to verify new probe sets are two-color odd and even sets. So let me explain how that works. First of all, you want to always validate and verify your sets starting in adherent cultured cells, because that's the easiest sample type to acquire really good images in. So you're going to want to choose a standard cell line that's super easy to culture, has low background fluorescence. I should pause and just explain that a little bit. Every type of cultured cell can behave a little bit differently in terms of just endogenous emission of background fluorescence. So you're going to want to avoid cells that have already a high level of autofluorescence that would be due to the presence of like collagen and elastins, cyclic ring compounds such as NADPH and riboflavins. Aromatic amino acids can cause that. A lot of cellular organelles, such as mitochondrial rich cells or cells with a lot of lysosomes. In fact, cells with high lipofuscin content, which is actually the breakdown that occurs in the lysosome, those cells like liver, kidney, heart, retina, and some brain cells have notoriously high levels of autofluorescence. So do yourself a favor, and when you're validating your probe set, start with adherent cells that naturally have a very low level of endogenous autofluorescence. And then order your probe set for the first time. If you have 48 probes, split it into 2 sets and have all of the even numbered probes labeled with one fluorophore, all of the odd numbered probes labeled with another fluorophore. And then when you stain your transcript, they're going to overlay. The evens and the odds are, of course, going to tile along the same transcript, and you'll get one overlaid punctate signal. We've offset that in this picture so that you can see all the evens were in red, all of the odds were in green. When you're actually seeing that not offset, you're going to have the color yellow because they're all overlaid with each other. And then some other things that you can do, in addition to the 2-color odd and even in a cell line, you can try using your same set in a different cell line that has known different expression patterns of your target. You can alter the expression of your target, for example, with drugs, to either upregulate or downregulate. And those upregulations, that differential expression pattern in response to the drugs, are going to be reflected in your Stellaris data. So that determines, that it's very specific and sensitive. You can also compare the data that you get with your spot counting from the single molecule RNA FISH technique, compare that with qPCR data, or you can try validating your probe sets in knockout cell lines. You can either have a conditional knockout or a stable transgenic knockout, and any sort of expression that's being detected by your probe set would disappear then in the knockout cell line. So it's very important to validate your cell lines. Looks like
All
we
right,
have
thanks for sharing the tips. Yes,
Sure.
we now come to the third poll questions, and we can to understand, what techniques do you wish to use Stellaris for? Do you wish to multiplex with Stellaris RNA FISH only, or do you want to multiplex with immunofluorescence, or do you want to multiplex with flow cytometry or any other techniques? And feel free to share your answers here, and we'll be revealing the answers in a few more seconds time. Cool, looks like majority, like I say, 2/3 of the audience here are very interested to multiplex with Stellaris RNA FISH and immunofluorescence. We got a roughly equal number of the attendees who wish to multiplex with only Stellaris, and another 38% wish to multiplex with flow cytometry. And I believe Julie will be sharing some of these exciting applications later on.
Great, thank you so much. We move on. I do now want to start sharing some of the exciting applications. Hopefully, you're all excited to get started with Stellaris, and you know that it's a very straightforward technique. And now let's show you some examples of some of the things you'll be able to do with it. The first is just basic mRNA detection. And I'm showing a gorgeous example that uses one of our control probe sets. This is the glyceraldehyde 3-phosphate dehydrogenase, or that GAPDH that we all know is a control workhorse. It's usually got very high expression, and it's great for just getting your protocol set up. It's a great positive control. And here you can actually see that the mRNA is present in the cytoplasm, and you can even see the 2 little transcriptional burst dots that are actually coming right off of the gene. So very specific. It's not good as a control or a standard for single molecule counting because the copy number is just too high to accurately count. Next, we're going to go into an example of what localization looks like with Stellaris. And here you're seeing TFRC is a very common cell surface protein. And like most cell surface proteins, it's synthesized while its encoding mRNA is associated with the ER. So the TFRC, if the mRNA is always associated close to the ref ER, then it should co-localize with something that is specific for the ref ER. And 7SL is actually a scaffold component. So here you can see in this example that in green, that TFRC is very co-localized with the red 7SL ref ER component, and they overlay to make that yellow color. So that's a great example of co-localization. Next, we're going to talk about how Stellaris can actually validate your knockout studies. And in this example, we're looking at HIF1 alpha in HEP2G cells. HIF1 alpha is actually upregulated in response to hypoxic conditions. So as you can see in this panel, at the bottom, we have our good old GAPDH positive control, and then we have the human HIF1 alpha upregulated, and you can see here the paucity of signal because of the short hairpin RNA knockdown. So Stellaris is a great technique to demonstrate these knockouts because for most short hairpin or even interfering RNA knockout, that knockout is transient. It's going to deplete the cytoplasmic mRNA, but not the nuclear RNA. And that's actually what you see in this panel. You see a little bit left over in the nucleus, but of course, all of it is gone from the cytoplasm, but that will eventually come back up and you can follow the trajectory. And that's just, as you see in the right panel, those results are verified by actually doing the gel analysis of that system. The next one that we're looking at is just an example of how Stellaris can be used to characterize cell lines. In this case, very common breast cancer cell lines, SKBR3, MCF, and MDA-MB. Morphologically, under light microscopy, they look very similar, but they do in fact have very different expression patterns of the HER2 receptor or ERBB2 mRNA. And they can be distinguished using Stellaris just on those expression patterns alone. Stellaris is great, as I mentioned, to look at alternative splicing. In this example, we're looking at the calumenin gene, which actually encodes two alternate calcium binding motifs. And depending on how specifically you target your probe set generation, you can target, in this example, we're using the Q670 for the common, what's conserved among all of the different variants. And then we have Q570 for exon 4. Exon 5, the fluorophore is calfluored 610. And by adding all of those three probe sets in a triplex, you can actually start to show what the expression patterns and the use of all the different splice variants under normal circumstances are. In fact, in this particular example that I'm showing you, this RNA fish was used as a validation for all of the RNA-seq data. So they just supported each other and added strength to the story of those alternate calcium binding sites. So I promised I would mention something about ice fishing. Ice fishing is basically targeting your exons and your introns. Here I'm showing an example from a oncogene MYC, and on the left panel you can see the exons that the probe set is targeting. you're going to see those because they're exons in the mature RNA in the cytoplasm, and you're seeing them at the transcriptional burst sites here, which contains the exons and the introns. The introns are going to be co-localized just to the nuclear foci. You should see 2 bursts for each cell. And then I do want to mention the flowfish. I know Joy had mentioned that a little bit ago, and that's a really exciting application of Stellaris for sure. It actually allows you to look at the expression of mRNA levels among many different samples, thousands at a time. I'm not sure how familiar you folks are with flow cytometry or FACS, which is the fluorescence activated cell sorter, but this is typical of the data that you're going to see in FACS analysis or flow cytometry. So in this particular example, we're using the adherent cell protocol because, not the adherent, I'm sorry, the cells in suspension protocol, and you're running it through the flow cytometer. You're looking at, in this panel, scatter plots of HeLa cells after hybridization with probes against CFOS mRNA, GFP, which is a negative control, and GAPDH, which is a positive control. The cells were left either untreated or treated with serum, detached, resuspended, and processed according to that cells in suspension procedure. The frequencies of cells positive for CFOS, GFP, which is negative, and GAPDH, which was positive, are reported above each gate. And as you can see, a really good representation of CFOS expression relative to your carefully controlled positive and negative panels. And I do also want to mention that there was just a recent paper that was published in, I think it was Science Translational Medicine back in March, that was using the HIV probe targeting several different HIV genes, and they got some really great results as well. There are a few things to consider if you want to launch into some of the flow fishing protocols. And one of them is the imaging platform. This is not, of course, a normal wide field microscope. We have a lot of customers that are very successful in this technique, and they perform flowfish using an ImageStream X Mark II imaging flow cytometer, keeping in mind that they have to upgrade the magnification packages because for Stellaris, you need either 60x or 100x oil immersion objectives, and most imaging flow manuals operate at 40x, so it is important to upgrade objective packages. Also, the extended depth of field used in these flow machines is important to consider because it allows imaging in more of a Z-stack than in a single plane of focus, like conventional microscopy. So depth of field is going to be incredibly important for the Stellaris application of this. And again, I can't stress enough, while you're embarking upon a journey of fish with flow cytometry, whichever probe sets you want to interrogate within that rubric of measurement, we highly recommend you first test their specificity and sensitivity in an adherent cell culture system. Once you've validated your probe sets, then you can move into the flow fish cytometry. We do have protocols that allow for rapid detection. A typical Stellaris RNA fish experiment can take anywhere from 8 to 16 hours, and in what's being termed turbo fish, they're actually able to shorten that hybridization period to as little as 5 minutes. I do want to point out that this does work, But as you can see from the inset with probe concentration, it requires an enormous amount of probes compared to regular fish protocols, sometimes up to 10 to 20-fold the amount of probes. So that's something for you to consider as well, the cost of each reaction. I do want to point out some of the technical advantages of Stellaris. The reason that it is such a powerful tool to allow for the quantification is because our spots do not use signal amplification. So our spots, I like to say they are better, they're just not brighter. As you can see from the cartoon, signal is only visible when greater than 25 unique probes tile along that transcript, and that's when you get the one punctate signal. Competitor systems for fish, their spots are a lot bigger and brighter. They're not limited by the diffraction limit, and there's an amplification step. So signal amplification, as you know, increases the risk of false positives and negative signals. So we eliminate those amplification steps and have the additional confidence when absolute quantification is required. So that's a big advantage. I'm really happy I have this image in here, knowing that a lot of you are interested in doing Stellaris with either sequential or subsequent immunofluorescent techniques. This is gorgeous data that's looking at HER2 expression. So the HER2 is being expressed on the outside of the SKBR3 breast cancer cell line, and that's being shown in this picture in purple. And then using Stellaris probes that target the actual mRNA from that, the ERBB2, you can see expression of the transcript inside. So this is actually really critical information if you're trying to design drugs that target different areas of genetic expression control, whether at the transcriptional and translational level or the actual receptor turnover at the cell membrane. Being able to simultaneously show data of both the finished protein and the mRNA transcript and their localization within the cell, and then up and down regulation is critical to a lot of people's studies. Stellaris is also a great adjunct in multiplexing data that you might need. As long as your microscope filters are set up for spectrally distinct fluorophores, you can multiplex 2, 3, 4 different colors at a time. And in this picture, you're seeing an example of 3 different targets, the EGFR in Q570, the POLR2A in CalFluoRed 610, and the topoisomerase 1 in Quasar 670. Those are very bright, stable dyes for us, and they can be used all in one experiment, and you can get great images of the multiplex. The products that we sell for Stellaris, we have the custom assays, which are assays that you design yourself. You get 5 nanomoles of the pooled probes. We have the ship-ready probes, which are great validated positive controls. I can't stress the importance of positive controls for these experiments enough. We also have the design-ready probe set, which are bioinformatically validated. A lot of the ones that are in our resource guide online have also been wet bench validated, but for the most part, these are bioinformatically designed by our team. We also have Stellaris buffers that are specifically designed for our protocols, especially if you're fixing cells with paraformaldehyde. You can see in this example how they reduce the background noise. And that's what a Stellaris experiment looks like. That's actually what you get when you order your probes. They come already blended and ready to put into the protocol. Speaking of protocols, we have all of them online, and our technical support team is here to help you choose the appropriate one, whether you're working in adherent cells or you want to multiplex or you want to start going into whole mount samples. And with that, I'll give you the tech support email address, and Dr. Kang's email is there as well. If you have any questions for her, please don't ever hesitate to reach out to my team and I. We love supporting Stellaris. And with that said, I'm happy to take any questions that you have right now.
Thank you, Julie, for this wonderful comprehensive overview. I know we're at the top of the hour, but we have a couple of questions and really want to address that.
Sure.
The first question is that as we understand, many of the attendees are working on FFP samples, right? So we have an attendee asking, working on FFP tissue or certain breed of mice. Are there any recommended probes or protocols for FFP tissues? There's one in the Stellaris website and also in certain published article for those protocols, but it does contain high temperature steps, including slight baking and permeation. So are there any possibilities that RNA is degraded because of high temperature during those steps? And also interested to understand what is the rationale of designing probes in the Stellaris probe designer in the website.
Yeah, so those are great questions. And absolutely, the success of any Stellaris experiment is based upon maintaining the integrity of the RNA transcript. And one of the difficult things is in FFPE, that's very difficult to do. So we do have a few tips and suggestions that can help with that. And if you can keep your sections as thin as possible, even down to 4-micron thickness, that helps with the permeabilization a lot. And while we do have a standard protocol that has the fixation and the permeabilization, there are a few different tricks that you can try with different types of fixatives and different ways to permeabilize. Permeabilization can be anything from 70% ethanol to using certain detergents like Triton X or SDS, and being able to, in a very systematic way, change one of those components and see if that helps with the penetration of the probes, or if a certain fixation helps rescue the RNA integrity, is something that we can easily troubleshoot. Because FFPE is a very difficult sample to work with, but lots of people have a lot of success with it. And to address the question about the probe design online, a lot of different factors go into the probe design. I did mention that it takes into account the GC content, it takes into account the TM, so that all of the TMs are matched for the probe set so that they're actually binding and working together. It's also why Stellaris is not appropriate for interrogating DNA targets, because that would be completely different TMs. The Stellaris probe designer also takes into account what's out there as far as the different species annotations, and it will ask you, are you working in human or mouse? And it'll start to screen against the actual model organism that you're working in. It asks about masking level, which is the amount of tolerance you're willing to have for potential cross hybridization, both in your species and in other species. It asks you about the space in between each one of your probes. If you have a short target and you want as many probes as possible, you'll maybe only have one nucleotide space in between those probes. But if you have a lot of room to work with, you may choose to have 3 or 4 spaces in between each individual probes. It does take into account the target length. We because of that diffraction limit I spoke about, where the probes line up and have to be within the 200 nanometers in order to work together and generate one punctate signal, any target that's over 8,000 nucleotides long will be too spaced out to get that diffraction limit and that good punctate signal. There's a lot of data on our website too about what goes into the probe design.
Thank you, Dr. Dre, for the detailed answers. We still have a couple of questions, but due to time constraints, we couldn't address every question, but rest assured we'll be able to deliver the answers to your email boxes. And really want to thank you everyone for joining us today, and of course for Dr. Julie Mangada for the really exciting introduction about this technology, and hopefully you'll find it helpful to help you with your research projects. With that, I'd like to conclude this webinar today, and thank you everyone, and enjoy the rest of your day.
Thank you, everybody.
Thank you, and see you next